So we left off with this discussion of T sub m, which is the melting temperature of DNA. And we said this happens normally about 85 to 90 degrees, somewhere in there, depending on the solvent, depending on the pH of what I've got to deal with. But in order for us in a laboratory to separate DNA, I have to make sure that I get it to the hottest temperature that I possibly can without completely killing it or disrupting it. The only thing I want to do is just rip those two strands apart from each other. So too hot is too hot, too cold is too cold, and the Goldilocks temperature is about mid 80 to 90 degrees, somewhere in there. So this started our discussion of being able to separate DNA into the strands, so that way we can take this strand and free up all of the base pairs on one of them and go in and make a copy, a complementary copy of that strand. So here's extra parts of the story. One, all of these base pairs that we're talking about, 3.1 billion. Not all of those are actually important. Now, when it comes to your genetic code, it is. Because this is what makes your DNA yours. What base pairs are present? What order are they coming in? All right, so let me write that over here to the side. Number one, which are actually there, present? And number two, what order are they coming to us in? Everyone's is slightly different. Therefore, everyone's 3.1 billion, there's a lot of choices in there, could be different or slightly modified depending on where we come from and who we got our DNA from. And we are a hybrid of those two people, our mother and our father. But to make you feel really bad, not all of these base pairs are important when it comes to body function. Because a whopping 2% are important. Wow. 2% of our DNA actually means something when it concerns our body and the functions that our body needs to do and all of maybe the RNA and the proteins that it makes. Only 2%? That's all it needs. The rest of it are just kind of lazy, good for nothing, mooching off of everybody. They're just there just to be there. And they're relying on the work of the top 2%. That's it. So we have a term for this. The 98% that does not do squat, doesn't contribute to anything at all, 98% are going to be called introns. Your body doesn't need them. It doesn't do anything for the body. No work is involved with that 98%. It doesn't contribute to anything. And the remaining 2% are actually the important ones. Those 2% is actually contributing to something that your body needs to do or needs to make. And this is called an exon. So I mentioned in a previous video that DNA, and I know we're only focused on DNA, and we're going to get back to just truly DNA stuff in just a minute, but I want to give you a bigger picture. The DNA's role is to make RNA. And when it does this, it really is only going to focus on the exons, the most important pieces of the genetic code, or basically the base pairs. And that RNA, in turn, is going to use that system of bases that it's going to see in order to make a protein. How does it do this? Well, the RNA reads the base pairs, and the base pairs in turn correspond to an amino acid, and the amino acid then will come together to make the protein. It is a roundabout systematic factory that happens here. But this is kind of where all of the orders come from. It comes from the DNA. And now we know that only 2% is actually important, and that 2% is called exon. So that 98% of the base pairs that you carry along with you in your DNA strand, it doesn't really mean nothing. It doesn't do a job. Now, could it do a job? The answer is maybe yes. Maybe. We just haven't activated that part of the DNA strand yet. And if we activate that part of the DNA strand, and your body understands that now it is an exon, and that exon should be done or should be used for a specific purpose, then maybe, just maybe, we can tap into areas of the genetic code that our body currently cannot tap into yet, or doesn't maybe want to tap into it. So there's a whole chemistry, there's a whole biological reason that this begins to happen. We can go through that steps if we want to, but that's more biology and biochemistry now. It's not really things that we need to worry with as far as the processes that go on in a laboratory. All right? But this is kind of where this is going to lead if we go down a different path. In addition to that, I told you that RNA will read the base pairs, and the base pairs will correspond to an amino acid. Typically, they are in triplets. Those triplets are in threes, of course. That's what triplet means. And those three basically are corresponding to the assembly of the amino acid to make the protein. Three options are out here that we call a stop codon. Codon, I kind of see the word code, and this is the code for the protein assemblage. And when it runs across these, the protein assembly stops. It gives the order for the RNA to stop making the protein. It is now finished. We do not need it any longer, and your body will stop producing it at that point in time. Now, let's kind of go back and refocus on the DNA molecule. So we have discussed this double-stranded DNA that goes through just like this. And there's base pairs in here that connect them. And we know those base pairs are ATs, Gs, and Cs. And we also know that the order of the base pairs are going to be different. So G, G, T, 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 A, C, G, C, C, A, T, and so forth. 3.1 billion of them are in me, are in you, in every cell. So I'm going to look at these base pairs, and the order of the base pairs are going to be pretty important. The reason is because in a forensics laboratory, this is going to help us determine whose DNA is whose. Bottom line. If I can figure out the order, or if I can figure out something about the order of them, then chances are I can be able to distinguish between two people's DNA sample. And what we do is that we use something that we call a restriction enzyme. And what happens is that a restriction enzyme is going to kind of sit on top of this DNA, and it's just going to kind of zip through and read it. Restriction enzymes, currently there's about 3,000 or more of them. They all do and read and do a job in a different way. Some of them, of course, are more common than others. But it really just depends on what you want to use and what type of job do you want it to do for you. And the restriction enzyme will read the DNA segment. And when it reads the DNA segment, it's going to be looking for specific areas. Those areas are what makes the restriction enzymes different. So let's say that this area that it's going to look for is TTT. Well, as it's reading the DNA segment, it's going to look and go, oh, look, TTT, ding, 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 ding. I can recognize that side. I can read that side. And what it's going to do, it's going to chop. Chops it into a piece. And this piece is what we call a fragment. So then it will continue on, read one base pair after the next, and it really isn't going to see any more TTTs that come around, right? No more. So I'm only going to get one fragment in this example. Now, keep in mind, 3.1 billion base pairs are quite a bit. So I'm probably going to get different fragments other than this one. Kind of makes common sense. But in this example, I'm just going to get one fragment, no more. So let's say that now I introduce a restriction enzyme that will come through and it will read a different area. And let's say this area that it's going to read at this point is going to be a GC area. Whenever it sees G and C together, it's going to go, oh, I'm going to stop. Now's my chance to do my job, and I'm going to chop, chop, chop. So as it reads the DNA fragment, it sees this first ding, ding, GC. I can understand that. I know what that means. So it's going to chop that DNA strand at that point. And then it will continue on, and it will continue to read. And look, it sees another GC. And it sees it, and it chops it at that spot. So I'm actually going to get one fragment from here, and then two fragments in between these cuts, and then maybe a third fragment or more, ... after it reads throughout the DNA chain. This is pretty important because, like we've said before over and over again, my DNA has a different base pair order than yours. Therefore, when I put a restriction enzyme on suspect number 1 and suspect number 2, these each have a different order of base pairs. And the restriction enzyme will cut it into different fragments, different pieces. Well, these fragments, they do have a molecular weight. I mean, we're talking about chemistry here. We're talking about carbons and nitrogens and oxygens, phosphorus. And we can calculate molecular weight for these fragments. That's not a big deal. The problem is that when we do that, sometimes these fragments are going to be very, very large. So they will have huge molecular weights that will come around. Well, to make matters a little bit easier, what we've decided to do is to talk about fragments in terms of base pairs. "50 base pairs long, 1,000 base pairs long, 10,000 base pairs long." Okay, so that makes my job a little bit easier to talk about the size of the fragments versus hundreds of thousands of molecular weights. We just term it in terms of base pairs, right? 1 base pair, 2 base pair, 3 base pair, 4 base pairs. That is what that's referring to. So now we need to get creative. If we can chop the DNA up into the fragments, we now need a way to separate them. And if we can separate them, we can look at what size fragments have just been generated. So how do we do this? Well, this is going to be accomplished with the help of electrophoresis. And this is how it's done. in a laboratory, if I wanted to take a sample of my DNA and I wanted to compare it to the sample of DNA that was found on the crime scene, I need to chop it into fragments and I need to see how it was chopped up. If it is the same way, same fragmentation pattern, then this is a very good sign that the samples match. Again, no one's will chop up identically. Unless maybe they're identical twins. So how do we accomplish this? It's actually kind of easy. What we do is that we take a powder, and this powder is called Agarose. Agarose is made with little sea creatures, pretty much. And this Agarose is going to be a white powder, and I'm going to dissolve the Agarose into a buffer. There's different types of buffers that could be out there. I'm not going to really be interested in the specific type of buffer here, but just notice a buffer typically includes a weak acid and a conjugate base, not DI water. And I'm going to make this solution. And this solution is going to be very clear. And I'm going to pour this solution into a mold. This mold is typically a 7 by 7 centimeter. I can get them bigger. I can get them much bigger depending on what I want. But typically a 7 by 7 is what we would see in a laboratory for our purposes. You might want them a little bit longer in case you have to run the sample a little bit longer so the fragments can separate out a little bit more. That's not a big issue. But normally a 7 by 7 is what I would see. And then what will happen is that this agarose will kind of gel on me. It will solidify somewhat, very much like Jell-O does in your refrigerator. Hot water, it dissolves the gelatin. When that gelatin dissolves, then sooner or later when it gets cold again, it will reform and it will wiggle. And that is our electrophoresis gel. Now, the way this thing is made is that normally we put what we call a comb up here at the top. And the purpose of the comb is to make a series of wells. And these wells will eventually get my sample. So if I'm looking at the gel from the top down, I am going to eventually see wells or areas that I would put my sample down into up here at the very top. You don't need a lot of sample. I'm going to take my DNA sample. It's typically going to be a liquid. I'm going to inject microliters. We're talking very little microliters here into those wells. And now I have prepared my DNA sample to be analyzed. Now, inside of these wells, we typically have what we call a loading dye. And the purpose of the loading dye is to increase the density of the DNA sample. That way, when I put my sample down into this jelly-like stuff, into the well, it will sink to the bottom. It will not get up and float out of the well because we're going to get ready to submerge this down into a buffer, down into a liquid. So I'm going to submerge the gel down into the liquid. It's going to fill up the wells with all of the liquid. Then I'm going to take my sample and then I'm going to inject it into the well, and my sample is going to sink down into the bottom and it will stay there. Well, if I took DNA and I have chopped, what will happen is that I'm exposing some of that negative backbone of the DNA molecule on both strands, right? If that's where I'm chopping. So these fragments become negative. And what I can do is that I can apply an electrical charge on this gel. If I start with the negative up here and the positive down here, I can lure those fragments down toward that positive charge. Negative and positive attract. But it's going to take some time. And these fragments are different sizes. And normally what happens is that the bigger fragments will typically stay up here at the top. So this DNA sample is beginning to move from the well, ... And then maybe this fragment can't really go anymore. It's big, it's large, and other things are going to start to run off and leave it. Poor thing. And then the other group of fragments continue to go down, and then maybe we get another fragment that kind of gets lagged behind. And then over a course of time, these fragments are beginning to separate from each other. And this is what we typically see. Each band represents a specific size of fragment that has traveled through the gel to get to that positive end. The larger base pairs are going to be up here at the very top. The lower base pairs are going to be down here at the bottom. And the reason is because those smaller fragments can move through this jelly-like material a little bit easier than the larger pieces. So normally what happens is that I have a sample that I will inject into the very first well. And in this first well, this is normally what's called a DNA ladder. And I'm going to get a sheet of paper, and that sheet of paper is going to tell me what size base pairs are present. And it will show me the order of them, and it will show me the base pair size for each one of the bands. That way I'll have that for my convenience. We're getting ready to do something with that. So then I'm going to take a sample of DNA. This could be from the crime scene. And maybe I want to skip a well. I don't have to use all of them. So I'm going to skip a well. I'm going to put it right in here. And then I have a suspect that I think that person did the crime. And I'll put it right in here. Okay. So I have these other two on the same gel. I'll load up my DNA ladder at the same time because it can run all of them at once. I need to run it all at once. And then I'm going to put that gel into this chamber. I'm going to turn the power on, and all of my fragments from the DNA ladder, from the DNA suspect, and from the DNA crime scene will begin to pull through that gel. Now, I might not see every band. This is just a ladder. It's just for reference. That's all that it is. But if this is the banding from the crime scene, if I have the proper suspect, then that suspect will also have identical banding, because these two DNA samples are the same. And if they're the same, they have the same fragmentation pattern. If they have the same fragmentation pattern, they're going to show me the same banding that happens on the electrophoresis gel. And we will call this a DNA fingerprint. No two peoples will be alike. Now, sometimes what they'll do is that they'll use 2 restriction enzymes, not 1. So they might process it with 1 enzyme and show you what that looks like and how it bands out. And then they might process it with a different enzyme and show you how that bands out. That way you've got 2 groups of comparison. And if both sets are pretty much identical, then that gives us even a better feeling about matching the suspect to the crime scene. This is what DNA fingerprint's all about. Now, we keep talking about crime scene, crime scene, crime scene, but parental DNA can also happen the same way. It's a little bit different theory, and you're going to do some of those activities because your DNA does not perfectly match your father nor your mother. You have your own. And half of your DNA will probably come from your mom, and half of your DNA will come from your dad, so it will be a conglomerate of the two. It won't be an identical match. That we'll talk about in a different video. But this is what we are referring to as our DNA fingerprint. It happens with enzymes. It happens with them cutting up into fragments. These fragments are getting loaded down into these wells up here at the very top. Over a course of time, these fragments are becoming separated, and as they become separated, then I can compare them. And if they are identical, then it's a pretty good sign that person did it. So what's the purpose of the ladder? Well, let's say that this band represents 1,000 base pairs, and this person, the crime scene, had a 1,000 base pair fragment, and this person kind of had a 1,000 base pair fragment too, right? But do you see how the left-hand side here is slightly up? It might not be exactly 1,000, but it's pretty close. So this DNA ladder can actually be used to make a calibration curve. And we can use the lengths and the distances that these fragments traveled in order to really pinpoint the exact number of base pairs that these samples have in common. So that's going to be talked about in a different video, and that's where I'm going to stop this one. So when we come back, we're going to pick up from this discussion. We're going to push on a little bit further into this concept of separating the fragmentation pattern.